Hello children, good morning. Welcome to the program NEET Foundation, and today we are going to discuss about part 2 of cell division and cell cycle. In our previous session we have discussed about the amitosis and mitosis. What is the introduction of the mitosis, who invented the cell division and the cell cycle processes and all. And in this process we are going to discuss about the third type of cell division is called as a meiosis, also called as reduction division, or also called as a heterotypic division. Okay, so it is called as, why it is considered as a reduction division? It reduces. What does it reduces? It reduces the number of chromosomes. In the mitosis, number of chromosomes will be equal in number in the parent cell and also in the daughter cell. But coming to meiosis, number of the chromosomes which are maintained by the parent cell. So as for example, like 2 chromosomes which are present in the parent cell, so it will be reduced into half, means one will be distributed towards to the daughter cell. So let us see here the introduction part. So this is the meiotic phases. In the meiotic cell division we can observe 2 phases, meiosis 1 and meiosis 2. In the meiosis 1, as like in the mitosis, so in the mitosis we can observe only prophase, metaphase, anaphase and telophase. But coming to meiosis, in the meiosis 1 we can observe prophase 1, metaphase 1, anaphase 1 and the telophase 1. And coming to meiosis 2 session, prophase 2, metaphase 2, anaphase 2 and the telophase 2. In the mitosis only 2 cells will be formed. Ultimately the 2 cells will be formed. It goes on doubling the number of daughter cells. But coming to the meiotic cell division, 4 haploid cells will be formed. A unique type of cells will be formed in the meiotic cell division. It is necessary for which kind of process? Mitotic cell division is for forming the somatic cells. But coming to meiotic cell division, it forms the reproductive cells. The meiosis, the cells which are undergoing the meiosis cell division, they are referred to as, those particular cells which are formed by the meiotic cell division are considered as gametes and spores. So this shuffles the genes. Inside the nucleus we can observe the genes. So each and every gene which is getting one from the paternal and one from the maternal. So there we can observe the crossing over, right? So this is a specificity of the meiosis process as compared to that of mitosis. The genes will be shuffled between chromosomes in a pairing process. Pairing process in the sense, at any sort of the regions, especially in the terminal regions of the chromosomes of the chromatids, we can observe the crossing over, so that the exchange of genes occur will be responsible for giving a rise to a new kind of characters. Nothing but the cells which are forming the new type of characters will be producing evolution. Nothing but the characters which are getting from the parent to progeny will be somewhat different than the previous generation. So here these are crossing over which are received from the parents, and it produces the chromosomes with new genetic combinations. So these new genetic combinations leads to the evolution by the varieties. Okay, so various genetic characters combinations will be produced by the meiotic cell division. And here the meiosis division produces genetically unique 4 cells. As just now I told you, ultimately the cells will be 4. Okay, so this is the meiotic cell division. Next, let us see the first phase of meiotic cell division, meiosis 1. In the meiosis 1, how many phases we can observe? Prophase 1, metaphase 1, anaphase 1 and telophase 1. So that is what which is shown in the figure, right? This is prophase 1, metaphase 1, anaphase 1 and telophase 1. Okay, so slight differences can be observed in the set of chromosomes as compared to that of the mitotic cell division. Let us see in detail what happens in the prophase 1. Within the prophase 1 we can observe 5 substages. What are those? Leptotene, zygotene, pachytene, diplotene and diakinesis. What are the phases here? Leptotene, zygotene, pachytene, diplotene and the diakinesis. So this occurs in the prophase 1. Next, Let us see in the leptotin what happens. Nucleus size enlarges. As like in the mitotic cell division, in the prophase, we can observe the disappearance of nucleus and the size of the chromosomes will be condensing. But coming here, we can observe the size of the nucleus will be increased and we can observe very clear picturization of the genetic materials, nothing but the chromosomes. So these chromosomes are divided as the chromatids are seen and which are helpful in identifying the chromosome. Identifying in the sense what is the shape of the chromosome, what is the structure of the chromosome can be easily visible in the leptotin structure. Okay. So this stage is also called as bouquet stage, it seems. Bouquet stage, nothing but, so it's a kind of crossing over stage. Okay. The chromosomes which are getting from the paternal and the maternal will be ready to exchange, join together and exchange the materials from one to another. The homologous paternal and maternal chromosomes come close together and at a particular point and appears. See here the paternal in the sense from the genes getting from father side and maternal in the sense genes getting from the mother side. So these two here we can observe the crossing over, right? So wherever we are observing the crossing over, so immediately we can observe those particular chromatids will be just crossed. After crossing, the crossed chromatid terminal will be getting the characters of a, if it is moving towards to the paternal to maternal, this chromatid of paternal will be moving towards to the getting the characters of a maternal, and maternal one will be moving towards to the paternal getting the characters of a paternal. That is the use of a crossing over. Okay, so see ultimately here this having the chromosome, the last terminal, one of the chromatid got a different structure, and even in the maternal side also one of the terminal of a chromatid also got a different structure. So this kind of crossing over which occurs in the stage called as a leptotin, first stage of the prophase 1. And next stage is the zygote. What is zygote? Here also we can observe the synaptonemal complex. We can observe the clear picturization of centrosomes with the centrioles. Further they will be forming as a spindle fibers. So here the bivalence pair of homologous chromosomes are being formed. Okay, so two chromosomes will be adjoining together and they are ready to exchange the materials by just joining together in a multiple regions. Let us see what are those multiple regions. Pairing of homologous paternal and maternal chromosomes is called as a synapsis. Where it joins, it's called as a synapsis. So through the synapsis, the materials will be exchanging from one to another, right? And next, so pairing starts at the ends, nothing but the procentric. Exactly at the terminal regions where we can observe the pairing process is called as a procentric pairing, and pairing starts at the centromere, the supporting region where the 4 chromatids of each chromosome is situated. Okay, so the pairing also occurs in the centromere and the random or intermediate pairing occurs at various places. First at the terminals, next at the middle region centromere, next at the random regions, everywhere. Okay, so here we can observe the synaptonemal complex. Okay, so this synaptonemal complex helpful for adjoining and exchanging the substances. So nucleolus enlarges and spindle formation initiated. Spindle formation will be initiated by the centromeres and the centrioles. Next one is the packetine. What is packetine? The third stage of prophase 1 of the meiosis 1. So 4 chromatids are seen in a bivalent. Bivalent in the sense 2 chromosomes will be formed over here. Okay, so see here each and every chromosome, but color will be different. Okay, so one from the maternal and one from the paternal, right? So see here the centrioles and the centrosome will be ready to form the spindle fibers. Packetine tetrads are formed. What are the tetrads? 4 terminals of 4 chromatids of each chromosome. So one of the terminal of a chromatid will be crossing over and ready to form a chiasma. What is chiasma? The point where the two non-chromatids in the sense 4 chromatids of one chromosome and 4 chromatids of one chromosome. Out of 4, one chromatid, one terminal, one chromatid, one terminal from paternal and maternal chromatids crosses over is called as a, that meeting point, junction is called as a chiasma, right? So, the points where two non-chromatids, non-related chromatids, this is a homologous and this is one of the homologous. But as they join together, so those are the non-related ones. So, adjoining at the chiasma, and at chiasma chromosome appear as a shape called as a X-shaped structure. As the terminals which are moving here, which are adjoining like this, so this point becomes a X-shaped structure. Okay, so that is called as a chiasma, which occurs in the stage called a pachytene. And next one is the continuous. Within the pachytene itself, here a very clear picture you see, homologous chromosomes. So here we can observe for the first chromosome, either the paternal or the maternal one, the 4 chromatids are being seen, and here also 4 chromatids are being seen. And next we can observe the chiasma, shape is X-shaped structure and tetrad condition of bivalent. Bivalent in the sense non-sister chromatids or non-related homologous chromosomes chromatids will be meeting at one particular point is called as a bivalent, and it forms as a tetrad. Okay, so the characters will be, the genes will be moved, but the terms will be, the terminal regions, chromosome sets will be 4 chromatids. Okay, and next one is the daughter chromosome. See here, what is the combination? One will be the blue and blue combination, 2 chromatids blue combination, and 2 chromatids, one is blue and one is pink combination, that is because of the chiasma. And one more is, one is pink and one is the blue combination, this is also because of the chiasma. And the last but not the least is the one of the arm, one side of the chromosome called as a homologous half pair of a chromosome. Half pair in the sense 2 chromatids. Okay, so this kind of process still occurs in the pachytene. And at last what happens after crossing? So what is the use of this crossing process? Why it is called as a reduction division? Why it is related to the evolution process? It relates to the origin of species. As the genes are exchanging themselves, so we can observe the new characters will be, variabilities will be evolved within the new generations, so that it leads to the origin of new beneficiary character species. So those particular species are survival of the fittest. With the beneficiary characters, they can adapt themselves to survive in a particular area. Okay, so crossing over exchange the genetic materials between 2 non-sister chromatids takes place, that already seen. So this results in recombination of characters and the linked alleles. The characters which are present in each and every chromosome are the linked alleles by the chiasma process. So after that, the generations will be produced by the crossing over. So this is responsible for the origin of a new species. So this origin of beneficiary new species leads to evolution. So by the evolution what happens? The ecosystem also will be benefited. Okay, so that is the use of the meiotic cell division. And next, last 2 processes are the diplotin and diakinesis. So in the diplotin process we can observe the spiralization. What is spiralization? DNA, the genes which are present in each and every chromosome will be spiralized. Spiralized in the sense they will be adjoining together, right? So let us see here, this is the diplotin process. The chiasma also we can observe. So the characters, the terminals which are changing, the sequence of base pairs also will be changing. Okay, in the DNA we can observe the base pairs. So those base pairs sequence if they changes, so they are getting, the offsprings will be getting the new characters. Okay, so this occurs in the chiasma process. So here we can observe the dissolution of synaptonemal complex, dissolution. Okay, so here we can observe the one terminal adjoins and one terminal looks like a X. Okay, and next stage is the diakinesis. terminalization, nothing but movement of chiasmata towards the ends of the chromosome begins. Terminalization, movement of chiasmata towards the movement of one terminal and the other terminal. One terminal, the crossing over finished, and the other terminal also has to be chiasmated. So that begins here in the diakinesis. So here what happens, the condition to the nucleolus, this nucleolus and nuclear membrane begin to disappear. Why they must be disappeared? For making the chromosomes to segregate freely within the cell, and that should be suspended freely with the support of a spindle fibers which are generated by the centrosomes and centromeres, right? And let's see the next one, next stage, second stage of the meiosis 2 called as a metaphase 1. So within the metaphase 1, the bipolar spindle apparatus is formed. Bipolar in the sense two polar regions with the spindle fibers. Okay, so within the spindle fibers, the homologous chromosomes are fused to form a chiasma or at a telomeric ends. So here we can observe the complete combination of two sets of chromosomes. Two sets of chromosomes are meeting exactly at a equatorial plate. As the stage comes towards to equatorial plate, the chromosomes exactly move towards to the equatorial region. In the meiosis 2, we can observe the crossing over of two terminals of non-sister chromatids will be seen, and the homologous chromosomes align along with the center of the cell with the support of a plate called as equatorial plate. So spindle fiber attached to the centromere. So where the centromere is, exactly attached towards to centromere. Why it has to attach? It has to divide equally. The centromere helps the chromosomes to divide equally and distributing towards to the another polar regions. So here the metaphase plate, center of the cell. So this plate plays a very major role in catch holding the spindle fibers towards to the centromeres and also chiasmata exactly near the terminal regions. Terminal regions, chiasmata is occurring exactly at the plate, and the centromeres are catch holded by the spindle fibers. So this occurs in the metaphase 1 of meiosis 1. And let us see here, these are the terminal ends, right? So these are the terminal ends, the telomeres, which are responsible for crossing over chiasmata and exchanging the genes. And let us see the next concept is the anaphase 1. Third phase is called as a anaphase 1. So segregation or disjunctions, nothing but, so previously the crossing over occurred and now the chromosomes, chromatids of paternal and maternal got the capacity to just move away from each other so that they can divide by the meiotic process in anaphase 1. So movement of daughter chromatids of each homologue. Movement of each daughter chromatid will be moved with the centromere. Centromere makes the chromatids to move in opposite direction. So previously they were in an equatorial plate and now they are moving towards to the opposite polar regions. Why they need to move? They have to form two cells, right? So these are the sister chromatids and we can observe the picturization. So blue color chromosome, one terminal, one chromatid will have a exchanging crossed over material with a paternal with the maternal and maternal with the paternal. And these genomes, chromosome sets are being separated. Okay. Next, the last but not the least in the meiosis 1 is the telophase. As like in the telophase of mitosis, we can observe the single cell will be dividing into two. First occurs the karyokinesis, division of the nucleus into two nuclei, and next we can observe the cytokinesis. Okay. The despiralization. So previously the spiralization occurred in the diplotin and after it was been followed by the diakinesis, and in this the despiralization occurs. Crossing over one divides. Okay, so as it divides, the nuclear membrane and the nucleolus, up till the anaphase stage the nuclear membrane will not be appeared, so that the equal distribution of the chromosomes is done in each and every set of the chromosomes. Chromosomes in the sense each and every cell. The daughter nuclei having each a haploid set of n of chromosomes are being formed. So here how many are being formed? It says 2 nuclei are being formed and the cells are also dividing into 2, right? So this is the last phase of the meiosis 1. Hope you all enjoying, right? Next phase, this is the interlinking phase in between meiosis 1 and meiosis 2. That is called as a interphase. What is interphase? The DNA again it goes for the synthesis process to get energy, right? So this interphase of meiosis, here we can observe the centrosomes with the centrioles for further formation of spindle fibers, and these asters are responsible for forming the spindle fibers and all. The nucleus and the nucleolus, chromosomes, single set of chromosomes will be formed, n number of chromosomes will be formed, and the nuclear membrane forms again, and next the cell membrane and the cytoplasm. So this stage is ready for undergoing the next stage of the division. After first nuclear division, meiosis 1 interphase occurs, and during which the cell prepares for the second nuclear division, nothing but the second meiotic cell division. That is what I was saying before. And now let us see the second stage of the meiotic cell division, meiosis 2. Exactly it is similar to that of the mitosis 2. So what happens in the mitosis? Prophase, metaphase, anaphase and telophase. So here also occurs same, it seems. What happens? So this, by the end of 4 haploid daughter cells are being formed, and it has designated as prophase 2, metaphase 2, anaphase 2 and telophase 2. Let us see in detail what happens in each stage. Yes, so a brief explanation I will give. So within this prophase 2, so we can observe the formation of the chromosomes. Chiasmated chromosomes will be again forming separately, and again in the metaphase 2 we can observe formation of the equatorial plate. So these equatorial plate, the centromeres of each and every chromosome will be attached towards the spindle fiber in an equatorial plate, and next forming the distribution of chromatids into the equal poles, nothing but opposite poles. And in the next anaphase 2 we can observe the elongation process of the cell is ready to divide, and here we can observe we can't observe the nucleus, but coming to the last stage of meiosis to telophase, we can observe the 2 nuclei are being formed and 2 cells are being formed by the karyokinesis and cytokinesis. 2 daughter cells already formed in the meiosis 1 at the ending stage of telophase 1, and other two cells are being forming in the meiosis 2 of last stage is called as a telophase 2. So ultimately 4 unique cells will be formed. So one will be the homologous set, and one will be the heterologous set, nothing but after chiasmated one. Okay, so this homologous and one more will be the heterologous. Okay, so this kind of different types of unique structured genes will be formed, so that the new cells also will be formed as a haploid number of chromosomes at the end of the meiotic cell division, right? So this is the end of this cell division and cell cycle. Thank you so much, dears. We'll meet in the next session.